Thank you, Norman, for that kind introduction. Thanks too to Ben Goodyear and the rest of the BASF team for giving me the opportunity of contributing to this discussion today. And most of all, thank you all for making time to join with us in this workshop. My topic is streamlining the path from topical formulation, innovation to manufacturing. The topics that I plan to cover are listed here. I want to first talk about the flow of activities from first articulation of product concept through to R&D. And then I want to talk about the various contributors to topical product performance, that is, the various characteristics of the product that need to be evidenced in order for it to be suitable for subsequent development. A theme of this workshop is quality, and so I want to discuss the manner in which activities, even early on in research and early development, can be geared in order to streamline the flow into subsequent development. A new topical drug product advances through a series of stages from concept through to commercialization. The initial phases of work, including formulation, innovation and optimization, are performed under good scientific practice conditions. Work in support of the safety profile of the product is done under good laboratory practice conditions. That includes, in our case, preparing materials that would be used for in vivo dermal toxicity studies, but also doing release and permeation tests in support of generic or 505J filings. Manufacturing of material to be used in clinical trials and for ultimate commercial use is performed under current good manufacturing practice conditions. The process is not always linear, but there are transitions between the stages, particularly a technology transfer at the point of the program moving into a cGMP environment. And that's the case even if all of the activities are performed within one organization. In Tioga Research's case, we're fortunate to be part of the Ncube Ethicals group of companies. And so a program that graduates from Tioga Research capabilities has the possibility of moving into development and manufacturing with Ncube. The final piece shown below are activities that neither Tioga Research nor Ncube directly provides as services to clients, namely in vivo work directed to preclinical safety studies or proof of concept, clinical work, but also support of regulatory aspects of the program. We're looking here at a more detailed schematic now of steps that a topical drug product program might take in progressing from initial planning through to filing of the IND. The color coding scheme is the same as on the previous slide. The green refers to general laboratory activities that we like to refer to as good scientific practice conditions, the light blue to GLP and the purple to cGMP conditions. The process typically starts with definition of the target product profile shown at upper left, and often in our work we interact closely with the client to develop that planning document. What follows is then product concept assessment and then potentially API selection if it's a new chemical entity, or selection of a source of API material, initially laboratory batch level and subsequently GMP condition. We are typically heavily involved then in pre-formulation, formulation verification and optimization work, and also the subsequent steps illustrated here including stability, container closure system, aesthetic panels, and I'll say more about each of these subsequently. I should point out that we function as a CRADO, a contract research and early development organization, and so we're on call to do whatever we can that will be of most benefit to our clients. In some cases, we're asked simply to make high quality measurements of skin deposition or delivery or permeation, but in other cases we're asked to play the leading role in advancing the program through this process to close to R&D. When we're asked to take on a program from the outset as opposed to contributing to just subcomponents, we like to start with a target product profile that presents the various characteristics that the product need evidence in order to be likely successful. These include the indication and the active, the administration route, which might be topical or transdermal or mucosal, and then the preferred formulation format and information about the intended dosage regimen and target strength, that's the concentration of active in the product. Delivery requirements are often not known precisely, but it's good to have an indication of how much of the active needs to be provided into or through the skin. The other characteristics are also important as they help bound the scope of the formulation innovation effort. A target product profile, TPP, does encourage this focus on the end product goals, which is beneficial throughout, and by documenting the goals, it helps provide a roadmap for our work on formulation innovation and refinement, and it also helps us structure the approaches that we will take indeed to formulation innovation and improvement. The experimentation agenda begins with pre-formulation work in 4 main areas: strategic guidance, that I'll say more about in a second, implementation and verification of analytical methods, experimentation preparation, where we're sourcing materials such as excipients and measuring solubilities, compatibilities, verifying study fluids. And then on a case-by-case basis, there may be other work. For example, if we're working with a peptide, we'll be concerned about degradation on skin contact, and we'll want to measure that before investing too much in other experimentation. A second theme of this workshop is digital solutions, and in common with many other technology areas, we're seeing increasing benefits from the deployment of e-tools. That is, various techniques as listed below that leverage data, information, and computation. In embarking on a program, we will typically understand the physicochemical characteristics of the molecules involved using molecular simulation techniques, and then apply natural language programming to interrogate the prior scientific and patent literature. We will take excipient information into account in designing our formulation innovation programs using databases such as the inactive ingredient database published by the FDA, but also other databases such as one of excipients used in cosmetic products that we have compiled. And we should also point out increasing utility of tools to help in excipient selection, such as Zoom Lab from BASF that we'll hear about later. A huge resource for us in future formulation programs is the vast databases of past formulations performance that we've accumulated over now 20 years in the field, and this resource just continues to grow in utility and extent as we move forward. As we embark on our experimentation campaign, we need to keep in mind that to be viable, the topical formulation needs to satisfy simultaneously multiple different requirements. Typically, we engineer our innovation and optimization campaign to focus first on the most challenging characteristic to realize. Usually that is achieving the appropriate level of delivery and permeation of the active. In other cases, though, it is the stability of the active, which may be susceptible to isomerization or oxidative degradation or hydrolysis. But even when we've achieved reasonable success against those metrics, the formulation needs to evidence the appropriate physical and chemical stability over the shelf life period and in the container closure system that will be chosen. It has to have the preferred formulation format and physical characteristics such as pH and viscosity. It has to have the appropriate aesthetic attributes. Those are the characteristics that the user or the patient feels when handling and applying the product. The excipients need to be acceptable and not introduce in themselves additional safety concerns, and the product needs to evidence the appropriate lack of skin irritation and sensitization. We're also sensitive, of course, to the cost of goods, and as we'll see later, the intellectual property dimensions can be very important, particularly when pursuing products along the 505(b)(2) pathway. Often the most challenging aspect of research and early development of a new topical drug product is achieving the appropriate level of delivery of the active or actives into or through the skin. To make these measurements, we use conventional diffusion cells, for example, of the design described by Thomas Franz in 1978, as illustrated on the upper right, where a piece of skin that may be an inch by an inch or somewhat larger is sandwiched between a donor well into which the formulation is dispensed, and underneath a receptor well that contains a buffer that provides sink conditions for the active once it's diffused through the skin. We've made measurements over the years on a broad range of tissues and various substrates. We make most of our measurements with human skin, but we've also made measurements on pig skin, various animal skins, nail, models of nail, sebum, mucus, mucosa, and other tissues as well. And we work with all of the formats that are well established for topical drug products, such as solutions, gels, lotions, creams, ointments, foams, patches. But also on the skincare side, there are various other formats, for example, rinse off or dry down formulations that we have also used. Most of our measurements are under finite dose conditions. Those are the conditions that under which we try to replicate the amount of material that's actually applied in the real world context. Although in the literature one will also read of a lot of work where an infinite dose of formulation is applied. And the analytics that we use are similar to those used by most other groups, primarily chromatographic techniques such as HPLC, LCMS, LCMS-MS. Radio label counting can be beneficial on occasion. And we've also, over the years, made measurements with some other techniques such as ELISA in the case of peptides. In a given program, we'll have a sense of how difficult it is likely to be to achieve the appropriate level of delivery or permeation based on the feasibility assessment that I mentioned earlier. The prime determinants of delivery and permeation are the characteristics of the active involved, predominantly its size, evidenced in its molecular weight, and its hydrophilicity or lipophilicity as evidenced in its log P value. However, when we put the molecule into a real formulation, all bets are off, and the extent of delivery and permeation can vary hugely, with even relatively small changes in formulation composition, as evidenced here by historical data from Manring and Smith at Procter & Gamble. They measured the delivery through human skin over a 24-hour period of the list of compounds shown here, which are all the hydrochloride salts of various amine functional drugs. They compared the delivery from a pure solution in propylene glycol to that in which 5% of the propylene glycol had been replaced by a well-known molecular penetration enhancer, or MPE. In all but one case, that was oleic acid. And they asked, to what degree does that small 5% change in composition change the extent of 24-hour permeation of the drug? The data are plotted on the right. We see in the most extreme case, there's a 450-fold increase in the extent of permeation. So that's the first thing. The extent of delivery can be enhanced very substantially by appropriate formulation engineering. The second point is that the extent of the same change in formulation composition varies hugely from one molecule to another. And this is usually the case. We are not able to predict what will perform well for a given compound based on data we have even for relatively similar compounds. This dependence of delivery and permeation on formulation composition is very well known. The literature is replete with examples of these types of phenomena, and it's very well known to people that work in the field. 20 years ago, when we began our journey in the direction of helping others develop new topical formulations, we were conversant with this, and we argued that as we can't predict how changes in formulation composition will affect delivery and permeation, we really need a more efficient way of making measurements. And so we began to prototype, to implement, to prototype, to verify, and then deploy in what we call production mode a series of high throughput experimentation platforms that would allow us to make measurements of how formulations affect and interact with the skin in manners that are more efficient than is possible with traditional diffusion cells. Some of the platforms that we've worked on are listed here. We take some pride in the acronyms that we've developed for each of these, such as Tornado shown below. These platforms, though, for example, Tornado, allow us to deploy multiplexed parallel arrays, essentially of miniaturized diffusion cells, and so we can make measurements of deposition, delivery, and permeation from libraries of formulations, and hence screen larger tracts of formulation composition space. And that allows us, or gives us, a better likelihood of discovering formulations that are high performing in terms of the delivery characteristics that we seek. Over the years, we've had a number of pleasing successes from the technologies that we have developed. An early one of these was a program we undertook when our team was part of Nouveau Research, when we were asked to develop an alternative to the Pensade formulation, Pensade being a topical formulation of the non-steroidal anti-inflammatory drug NSAID diclofenac. And elements of the target product profile are listed succinctly here. The intent was to treat osteoarthritis of the knee, the pain and the symptoms of that. Parent Pensaid comprises 45.5% DMSO, and the intent with these alternates was to have DMSO-free formulations, but they nonetheless needed to deliver substantially more of the active diclofenac, it's actually the sodium salt that's incorporated into the formulation, than the parent Pensaid, yet have short drying time, be a gel rather than Pensaid's free-flowing solution, have stability in the container closure system for at least 2 years, but also be patentable. As illustrated on the right, we deployed a early high-throughput experimentation platform, Tempest. We did some preliminary work with another platform to narrow down the space in which we'd make these more detailed measurements. And as illustrated, we were able to discover not one, but a family of formulations that all were able to deliver substantially more of the active through the skin than was realized with the parent Pensaid with its 45.5% DMSO. The patent claiming these compositions was issued in 2010. Our Nouveau Research colleagues challenged us to develop a follow-on product to parent Pensaid, for which the target product profile characteristics are summarized here. Importantly, it should be a gel in contrast to Pensaid's free-flowing solution, suitable for BID dosing as opposed to Pensaid's QID, improved aesthetics, similar stability, but a limited compositional change from Pensaid so that the safety data would translate more or less directly, but it had to be patentable. This product is now called Pensaid 2%. It was approved in 2014, and today it is the top-selling topical prescription drug product in the US. The prime inventor on this leading patent, Ed Kissack, was a co-founder of Tioga Research and serves as our chief scientific officer. Jagat Singh has been a consultant to Tioga, and Dominic Kingsmith is also a key member of the Tioga Research team. It is often said that formulation composition of matter patents are of limited value because the composition is readily circumventable. By historical development methods, that may be true, but Pensaid 2% evidences that there are occasions, if the work is done right, where formulation composition of matter patents can be very valuable. In this case, 6 generic companies filed paragraph 4 certifications claiming the invalidity of the patent. 5 settled out of court. The 6th took it all the way to court where the patent was upheld. They appealed, and under appeal the patent was again upheld, evidencing that formulation composition of matter patents can indeed be hugely valuable. We have found in programs like the ones I've just mentioned that high-throughput experimentation can be very valuable in establishing a sound basis for patentability, and in particular composition of matter patentability. The extent of delivery and permeation, or indeed stability in some cases, can depend sensitively on the formulation composition. Such effects are generally not predictable, and so the best formulations, if they're indeed novel, are non-obvious and then suitable for patentability. So broad-based screening can highlight not only the strong performers, but also that general landscape of other compositions that perform inferiorly to the optimal formulations, which provides further support for the IP position. And our methodology for doing such screening, that we call cascaded screening, in conjunction with our high throughput experimentation platforms, has quite consistently provided a good basis for patentability. I'd like to close by providing some thanks, first of all, to our fantastic team at Tioga Research, but also our colleagues at Topicals, who've been a huge help. They've been a long-standing client for many years, but recently they've been a huge help in implementing our GLP capabilities. But thanks also to our many collaborators, and particularly to our clients. We really enjoy working on the exciting new programs that our clients bring to us. There are huge opportunities for new topical products, and it's a real privilege to have the opportunity to contribute in even a small way to the furtherance of some of these new possibilities. Thanks also to former colleagues at F-Cubed and Nuvo Research, and thanks also again to all of you for making time today. With that, let me pass the baton over to the subsequent speaker, Professor Narasimha Murthy from the University of Southern Mississippi, who's been doing work in this area of skin-applied formulation science for many, many years. And most recently, he's been helping the FDA think about best practice in terms of doing release and permeation tests under GLP conditions. Dr. Murthy.